All right, welcome everyone. We're going to get started. So thank you for coming to today's seminar. I'm pleased to introduce Dr. Theresa Swift-Scanlan, who is an associate professor for the School of Nursing and is also the director of the School of Nursing's Behavioral Research Lab. Dr. Swift-Scanlan's research is focused on epigenetics of chronic illness, specifically to identify molecular biomarkers that can be utilized to improve risk assessment, early detection, and/or targeted interventions for cancer and neurodegenerative disorders. She holds a patent that's recently licensed by Swift on a method that quantitatively assays epigenetic changes in clinical samples, particularly those with limited tissues or cellularity. And she routinely integrates omics data with candidate gene data derived from mid-plex platforms like Massarite, and she was successful in obtaining the first NanoString platform in the Commonwealth, which is the topic of today's lecture. So join me in welcoming Dr. Swift-Scanlan.
Thank you very much for that warm introduction, and thank you all for attending today. Just by way of explanation, I was exposed, turned on to the NanoString about 5 years ago. I do a lot of work with archival tissues. I don't know if anybody here works with formalin fixed paraffin embedded tissues or difficult clinical and biological samples that have just limited amounts, limited tissue, limited cellularity, and was really taken by NanoString in terms of some of its capability. With that, I have a disclaimer that a lot of the images, the lion's share, have been taken from NanoString's, some of their PDF materials or PowerPoint materials. So it looks like I'm an agent for NanoString. I am enthusiastic about the platform, but I just want to give you that that disclaimer, and also a shameless plug here. Let's see, how do I advance? Just with the mouse, or just
you should be able to just use the
I thought I did, but it wasn't.
click on that.
I'll try. Hey, yeah. So anyway, so we have the first NanoString in Virginia. We just got it in the last year. Who knows? They might have others that are at the competitors a few hours away in the mountains at UVA. So without further ado, and I know that some of the folks that I'm speaking to here today and online have different backgrounds, and I think from talking to Amy that. Can you see my cursor when it flies over here? Okay. That, a lot of the bioinformatics and some of the technologies that you guys have been exposed to, whether it's microarrays, RNA-seq, next generation sequencing, et cetera, almost all of them are based on amplification, on PCR-based methods, and or a combination of conversion. So you're converting the original RNA to cDNA, that sort of thing. Similar idea for those of you who do epigenetics work or epigenetics research. Almost all of the platforms, there are some exceptions, but whether they're candidate-based platforms or omics platforms, they rely on an initial conversion of the DNA sequence via sodium bisulfite treatment. So basically what this little cartoon here illustrates is that when you start off with any sample and you have these CpG dinucleotide sequences, which is what you're measuring methylation at. So for the 850, have you guys gone over the 850K beta arrays? Not yet. Okay. So some of the platforms out there, genome-wide, will assay 850,000 the differential methylation for 850,000 CG dinucleotides throughout the whole human genome. The previous platform through Illumina was the 450K beta array, and like the name says, it interrogated about 450,000 CG sequences. So, so again, whether it's omics-based or candidate-based, you start off with a cytosine that's methylated, and if you treat it with sodium bisulfite, it's still methylated at the end of the day, so there's no change. And again, this also relies on not only conversion but PCR. So say in another example, at low
If everyone could please mute their microphone who's online, thank you.
You start off with an unmethylated cytosine here, and when you treat it with sodium bisulfite, it becomes a uracil, and then in the process of PCR, it becomes a thymine. And so in this way, we irrevocably, whether it's we're taking RNA to cDNA, we irrevocably change the sequence, and then we make inferences about what was original in our sample or our clinical sample, for example. And so I give some of those examples down here. So in contrast, in terms of the technology, NanoString uses direct digital counting. So I'm going to talk to you a little bit about how that works. Can everybody hear me okay? Yeah.
Yeah, in a way, it seems like you have more than
Okay.
Oh, sorry.
Okay, so Amy has muted you guys because we were hearing you in here, and it was kind of entertaining actually. A little more entertaining than I am. Okay, so I'll just close out of this guy. All right, so some of the advantages of NanoString, as I mentioned before, it does have biases, and I'll mention what those are. But from all of those other technologies that many of us are either working directly with, or we're certainly utilizing the data derived from those methods, there are no enzymes involved. And as I said, there's no amplification, and by and large, there's no conversion step unless you want to incorporate PCR into your analysis, and I'll get into that later. As I mentioned before, I got sold on the platform because it works really well with archival tissues, and these are tissues that are fixed routinely in formalin, and then they're embedded in wax or paraffin, and slices of those are made. We extract nucleic acids or proteins from those slices, and also we stain them with proteins so that histopathologists can go in and say, oh, okay, well, that tumor is positive for the estrogen receptor protein, for example, or negative for that receptor. And what happens with formalin fixed tissues is that, and some of you again already know this, but the formalin heavily cross-links the nucleic acid and often shears it. So by the time we start trying to work with it in the lab, we've got little tiny bits of nucleic acid, relatively speaking, and it's not in such great quality. Again, harping back on this idea of many of the other methods use amplification. In order to use amplification or PCR, you have to have a sizable chunk of nucleic acid. You know, for many of the technologies I mentioned previously, you know, you really want something in the at a minimum 200 base pair range, and certainly if you had chunks in the 500 base pair range, that would be something that you would want. And because NanoString doesn't rely on amplification, that means that it actually works wonderfully with these very difficult tissues that people have really struggled with. So again, the precision comes from there's direct digital counting of these fluorescent code tags. Again, this is sort of the plug of using some of their advertising. They do offer, and I'll get to this in a moment, a suite of analytics packages that are actually, that seem to be pretty comfortable. Where I'm still in the process of learning them. But I certainly would also suggest collaborating with biostatisticians and bioinformaticists as well. I put a star here in terms of this as an advantage. Certainly it's an advantage that it's highly multiplex. So you can be looking at 800 targets, up to 800 targets, and this could be 800 messenger RNAs, hundreds of microRNAs, and most importantly, it could be a combination in the same sample from the same person of looking at, say, 770 unique messenger RNAs and 30 unique proteins. So that's incredibly powerful. The reason I put a red star next to the 800 targets is, you know, for people that are used to the space that it occupies in terms of technology, NanoString is not going to be like RNA-seq. It's not going to be giving you information on 22,000 mRNAs or 25,000 mRNAs in the human genome. It's going to be up to 800 targets. So that's the kind of space that it occupies. So certainly if you've, if either you or in the literature you already have omics data that you can kind of use back and forth, sort of synergize those data with more candidate data that you're interested in, or if you have, again, samples that you're just saying, hey, 800 is enough for me. If I found, and I knew that there were genes on that code set that or pathways that I'm interested in, that's also something that you could do. Okay. I guess I need to, if I click off, it doesn't work. Okay. So it works well with a lot of different sample types, as I mentioned, especially with cells of limited cellularity or quality, but it works with just about any sample type you can imagine. NanoString has capability for looking at single cell expression, copy number, so at the DNA level, copy number variation, SNPs, fusion genes, of course, gene expression through messenger RNAs. They also have microRNA panels, long non-coding RNAs, and then, as I mentioned before, you can do what's called some of their 3D or three-dimensional technology, basically in multiplex, looking at proteins and RNA and SNPs all together in the same sample. All right, so what this is based on in terms of the core chemistry, and sorry about this blurry image that I snatched, it ended up not having much resolution. But basically, what you have is this chemistry that was developed in the Institute for Systems Biology in Leroy Hood's lab in Seattle. And so it relies on this fluorescent barcode. So you have a total of 4 colors, green, red, blue, and yellow. And those are aligned in a chain of 6 different colors. And so if you were to do the math on that, you end up with about 900 possible color code combinations. The reason the limit of the assays up to 100 are used is that there are always positive and negative controls and housekeeping genes that are included in each code set. So in terms of the genes of interest, it would be roughly about 800. So what's going to happen, and I'll explain this in a moment, this tag that you have is going to hybridize directly to a nucleic acid of interest, and each target that you're interested in is going to have a unique combination of these color codes, and at the end of the assay, there's a really powerful camera in the analyzer, and it takes a picture that looks something like this, and it's a simple counting. For those of you who have used fluorescence in the lab, it's taken me a long time to get this. Like, I keep thinking, oh, it's measuring intensity. It doesn't measure intensity at all, and that's also one of the beauties of it. It's just a simple counting, so you don't have to worry about biases that could occur from different fluorescent intensities from background and that sort of thing. Okay, so basically the platform is fairly straightforward looking. You've got this one instrument called the prep station, and basically a lot of the chemistry is done on that. The first thing that you do is hybridize. Typically this is depending on the assay, again, whether it's RNA, some type of RNA, DNA, protein, or a combination thereof. You hybridize overnight, and then on the prep station here, this is just a picture of what it looks like inside. In essence, you put all of your reagents that you need, the code sets and everything. You have all the tips that you need, the sheets, tube strips here where you have your samples. And then a robot spends about 5 hours going through this once the experiment starts. This is actually critical because once it starts, you really can't stop it. So you want to make sure everything's in place before you do that. And then here is where you have a cartridge. And this is where all of the samples end up. And I'm going to pass these around for you guys who want to take a look at it. I'm not sure exactly the date. When I first became aware of NanoString, I think it was around 2012 or so. It hasn't really been around that long, but some of the different platforms have changed. This used to be this is an older cartridge and has information here, 12 channels for 12 different samples, again, where you'd be getting information on 800 genes of interest. And now the cartridges that we use are about this size, but it's the same idea. So I can pass these around to you guys if you want to take a peek. Yeah, so that's pretty much that. The cartridge that I've just shown you, the small one that you'll be seeing, fits into here, and there's an electrode assembly, and I'll talk to you about that in a moment of what that's used for. So NanoString has a number of off-the-shelf assays, but you can also make, in working with NanoString, you can make any custom assays that you're interested in. So regardless of whether it's NanoString doing it from an off-the-shelf assay, the process is absolutely the same. You go in and grab, say we're interested in beta-actin, for example, you go ahead and grab the sequence from NCBI. You would choose, you choose basically about 100 base pairs or so, and you do all the stuff that many of you who do this work are familiar with. You'd look for secondary structures and synthesis issues and the uniqueness of the sequence against transcriptome and system controls, all that sort of stuff. So then the next step is you take that 100 base pairs of sequence, which is right here, and in essence, you basically synthesize oligos, cut that sequence in half for all intents and purposes. And you'll take one part of the sequence here, is going to go to the capture probe, and the other is going to go to the reporter probe. And so the way that this works is, and I think that this is not exactly correct, but forget that fluorescence barcode says up there. I don't know how that happened, but one part of that 50 base pair sequence is going to bind here to this probe, and it's going to have biotin on the end. And that biotin is then going to bind to the cartridge that I'm sending around to you that has streptavidin coated on it. So the biotin, that's what's going to happen on that end of the capture probe. The target probe here in purple, this is what is going to be this unique reporter probe sequence that I've talked to you about. And the other 50 base pairs of the gene of interest is going to bind to this. So in essence, that's how it works. And so then what happens is when you expose these probes, both the capture probes and the reporter probes, you basically hybridize these to your sample of interest, and it binds your target mRNAs, and then those get, in the prep station, those get each sample, each of 12 samples gets transported to a unique well on the cartridge. Okay, so the barcode chemistry, I'm going to be focusing on messenger RNA now just as an example, but I did want to briefly say that basically the barcode chemistry uses RNA or DNA detection. Basically it's using an endogenous nucleic acid, and that's how SNPs, copy number variations, and so forth, and messenger RNAs, microRNAs, et cetera, can be detected. On the protein side, basically what you end up having is a unique single-stranded DNA tag that's synthesized here to a primary antibody. So this is how you're able to detect the proteins, but with the barcode chemistry, again, the idea is the same. You could have a code set where you had 770 mRNAs, and then you could have 30 proteins that you have unique code tags on them. So that's how you're able to actually interrogate 30 different proteins and 770 different mRNAs in the same sample. And again, without going into too much detail, the microRNA assays are more complex, and you could spend a whole lecture actually talking about them. Again, sorry about the resolution of this picture, but I just wanted to sort of globally summarize that from RNA, you can look at, messenger RNA, as I've talked about before, with this same idea of a reporter probe and a capture probe on this side to your sequence of interest, to your RNA. The microRNA, there's some pre-processing that has to be done, and basically there's a bridge because the microRNA oligos are so short, and you have to develop a bridge linker to join them together. But again, the basic idea of the barcode chemistry is the same. And again, basically it's just variations on a theme. The same thing for how SNPs are detected and copy number variations are detected. And I already mentioned to you that single-stranded DNA that is able to link to the protein of interest. That single-stranded DNA, of course, then is linked back to a unique reporter probe barcode tag. Okay, so the first step in the process is you'd hybridize, and typically for most of the assays, this is done overnight. So say in this cartoon, you've got all of these messenger RNAs running around in your sample, and you want to find out what they are. And for your code set, you have each of the code sets is going to have for each gene or each messenger RNA is going to have its own unique sequence of colors here. And so you're going to have your capture and your reporter probes during the hybridization reaction. The next step is they're going to bind to each other, and then you're going to remove the excess reporters here that are unbound and the excess probes and wash them away. And then this is the really cool part. So that capture probe that has the biotin that I mentioned to you, it's going to bind to this surface of the cartridge, and this is what's I'm sending you around now, this particular cartridge. So then the biotin grabs here and grabs to the surface. And the next step is really kind of cool. Have you guys seen, I never understood where these things came from, but at car dealerships or used car dealerships, you see those big balloons that have like a really tall guy that's, I don't know, 20 feet tall and has little arms and it's got air going through it and it sort of flops over, right? So this could actually happen because ultimately a camera is going to read each of these codes and count them. And so in Leroy Hood's lab at the postdoc, and I don't know the person's name, but what they did was really ingenious. First they developed this, and then they figured out a way to immobilize all of these codes and linearize them. So that's the name NanoString, which is in that electrical current, there are electrodes in the prep station that I showed you earlier on. So in this electrical current, all of these guys lie down in a linear fashion so that they're not overlapping each other and so that they can be counted. And so basically what you have here, it just ends up being a simple count. The camera images the surface. And so again, one reporter code equals one messenger RNA. This is sort of a cartoon of the cartridge that I'm sending around to you now. It has 555 regions or fields of view times 4 color channels for each region, and so you end up getting 2,200 separate images. All of those images are then processed during the analysis step, which takes about 5 hours. Typically, we do that overnight in the lab as well. So you collect this image. Of course, it's not in color yet, but to the camera, it's going to look something like this. And this is kind of cool. So what they do is they have these in this universe here in this galaxy of barcodes. They have here this like a multispectral bead, and it's used as an alignment fiducial. So there's software correction that NanoString developed that can reposition those scan fields to ensure that spots are in recognizable order. So it would look something like this. And it totally linearizes them so that they can be read. So again, in color, it would look something like this to the camera. And so basically that's what happens. It's just a simple count. It doesn't have anything to do with fluorescence intensity. So if you look at this combination here, again, they're blue, green, red, yellow. There's 3 of those. So for gene X, there would be a count of 3. There's only 1 of this. So for gene Y, that would be a count of 1, and so on. So these codes are then counted and tabulated. And basically, you get data that looks like this. So this is real data output for a particular experiment that had different treatments. So for example, for this particular gene, you can see that across these 3 treatment groups, the number of counts for that mRNA varies and then you can do various types of statistical analyses on that. It's just simple counts. The dynamic range is very wide, up to 5 logs. It's highly reproducible. And what's really nice about the counting method, with the proviso that your binding density is okay, and I'll get to that in a moment. These are things that I'm learning as I'm more hands-on with the technology. But you get sensitivity and precision even at very low expression levels. So some genes in your sample may be important, but yet they're expressed at pretty low levels relative to other genes, and you can assess that out during the analysis. Roughly 70, just sort of on average, 75 counts is equal to about 1 copy per cell. So again, you can use, we haven't done this yet, but NanoString stands by that you can use whole cell lysates, basically use that directly as your sample without extracting nucleic acid. Blood, saliva, urine, human tissues, plants, microbes. A lot of folks working in microbiome studies are using this technology as well. As I mentioned, for DNA, you're getting information on SNPs and copy number variations. For messenger RNA, up to 800 genes. MicroRNAs, typically, we're in the process of actually running a microRNA experiment from exosomes now. I believe on the panel that we use, there are about 400 or so microRNAs, so not all the assays have up to 800. I just wanted to make that clear. And also, they also have mixed microRNA and messenger RNA, what they call their merge panels. So again, with proteins, the number of proteins that NanoString has actually, basically they get the antibodies and they develop code sets for those particular proteins. And I think to date, they've worked up about 50 of those proteins. And I know you guys might have some questions about that later. You can also work with NanoString with the proviso that you know that you have an antibody that you can trust, that's commercially available, and then they can label it for you with your unique single-stranded DNA and its unique code set. Okay. So again, all of this is just by way of review. It does really well with both translational and clinical studies, obviously for basic science as well. But in terms of, I think what makes it great for translational is just its flexibility in terms of dealing with many different types of clinical samples. Okay, so the analysis suite comes from NanoString. So what would happen is if you guys, and I'll give you my information at the end and my card, and if this is something that you're interested in, feel free to contact me. But basically what you do is you go to the NanoString website and you register. And then after doing that, you'd be able to download the software. So after you get the files, and I'll go into that in a moment, the files from the analyzer, after the cartridge is read, you get two main file types. You then would open those files in the software, launch those, and then you'd run sort of customizable normalization and fold change measurements on there. So they sort of set up this suite of analysis that kind of do it yourself analysis. But and again, we're working with this, but NanoString, I think, would say that, oh, we don't need a biostatistician or bioinformatician, and I think that's I wouldn't want to do away with people like Amy. I love people like Amy and, the people that we have here on campus and other collaborators that you have not, outside of VCU. And I'm looking forward myself to working with some of those folks as we take deeper dives into the data. But certainly from a convenience perspective, speaking from someone who's worked with omics data, you get a large amount of data, and just generating heat map images or things like that is not necessarily trivial, and at least at first blush and first pass, you can get some pretty nice graphics out of this. So I think I think that NanoString has done a good job with that. So again, depending on what biological questions you're asking, a lot of the advanced analysis modules are based on R. And they're compatible with a lot of analysis programs, including, you've probably gone over some of these, Amy, the Ingenuity pathway analysis, different types of omics suite packages, and so forth. So these are just some of the outputs and I've generated some of these just on data that we've been processing in the lab. But you can, get box plots. Certainly you can get clustering and heat map analysis, and you can do that on raw data. You can do it on the data that you've normalized, scatter plots, volcano plots, that sort of thing. And then it will even return some information on pathway analysis. But again, I'm still in the process of learning myself. For me, I think it would also be helpful in working with someone, or if you're comfortable in working with, Gene Ontology or David or some of those other platforms, to be able to take your data, which you absolutely can. It's just in counts, and take it and analyze it yourself, or with whoever you're collaborating with. That you can certainly do that. You don't just have to rely on the suite that comes with, but it's certainly nice to have this right there, so that once you get used to the process, you can really start looking at your data and going through it. So again, I think I just added this slide again, or added a little bit more to it. In general, the input of the DNA and the RNA depends on the assay that you're doing and also the sample type. So typically, if you're working with FFPEs, you're going to want 200 to 300 nanograms of input of nucleic acid, certainly for the RNA. And the reason is it definitely is highly precise, but like I mentioned before, a lot of the RNA that you're going to get from formalin-fixed tissues is going to be highly degraded. And so in a sense, you're going to want to overload. And I see Megan in the back. We're working with her and Jennifer Kablinski on doing a microRNA experiment from exosomes that they've isolated. It's hard to believe. We'll believe it when we see it, and hopefully we'll have results for these guys in the next month. But exosomes are supposed to just be chock full of microRNAs, and so it's like you're just trusting that all of this is going to work when you load a very small amount of total RNA, like 3 nanograms per microliter, and maybe you're going to load 15 nanograms of this and just kind of hope that it's going to work. So we'll see, but obviously there are papers that have been published that have shown that this does indeed work. Laser capture microscopy, single cell analysis. Again, I mentioned some of these. Circulating tumor cells, and exosomes. So lots of possibilities, I think, with this. I did want to show you some data from NanoString, that this is basically showing really high R-squared values between total RNA that was purified from cell lysate versus the cell lysate itself. So you can see that it's highly comparable. And again, I sort of alluded to this, but there are a number of studies that have been done out there showing a very, I think impressive, 0.85 is decent, 0.83 correlation between fresh frozen specimens and FFPE specimens from the same individual. This is just looking at the particular genes that were in this assay, and looking at the correlations between, again, fresh frozen in green here and FFPE in black. So I mentioned before that it's not just the off-the-shelf assays that are available. You can also customize depending on your organism of interest. So that just shows, whether it's insects, plants, microbes, mammals. If you can get nucleic acid from it, then you can do the assays. And these are common species that, so you can not only multiplex targets, but really can multiplex species. Obviously, a number of those have unique sequences, species-specific sequences that that you can develop custom code sets. In fact, some of that already has been done with NanoString for microbiome type studies. Okay, so this just gives you a quick idea of some of the off-the-shelf panels, and I just lot of cancer and cancer immunology panels in humans, and you can just see some of these here. They also have immune profiling panels. So again, this would be 770 messenger RNAs for the mouse. They also have microRNA panels. These are, again, are off the shelf and ready to go for human, mouse, and rat. And then here's what they have available, again, in terms of off-the-shelf CNV types of analyses, as well as a karyotype panel. So the actual genes on all these lists, again, are available once you register on the NanoString website, so you can really look through a list of 800 genes and say, okay, does this have what I'm interested in? And what's nice about this, too, is that say you say, okay, I really want this pan-cancer or breast cancer 360 panel, or pan-cancer immune panel that has 770 messenger RNAs. You can then, and if there are proteins that you want to look at the same time, you can do that. But you basically got 30 slots. So you've got 770 plus 30 question mark is equal to the magical 800 number. So then you could actually add in, you could design yourself, just like I showed at the beginning of the lecture, messenger RNAs that you're really interested in your particular world, in your particular sample, and the biological questions that you're trying to answer. And so you have those 30 slots, the capability of being able to fill in and amend those panels. So I think this is just another slide basically saying the same thing. And a couple of the panels that they sell already come, how can I say this? They come with an analytical capability where you can get returns on certain pathway analysis that's proprietary. So, for example, I work with breast cancer data, and for those of you in the breast cancer world, the PAM50 gene panel, the minimum 50 gene set that defines the 5 major subtypes of breast cancer, was developed in Chuck Peru's lab at UNC. And working with NanoString, that basically that's been patented now, and that's called the ProSigna panel. So when you buy that particular panel, it's actually more expensive because the software that enables you to analyze that data gives you a return on answers to, say, whether the tumor's hot or cold from an immune perspective, that sort of thing. But overall, there's, in terms of looking at the hallmarks of cancer for people that work in the cancer world, you get these TIS or tumor immune signature measures, but they also, these sort of 360, and the reason they call them 360 panels is they have overlapping reference genes, and they can also be compared for, like, basically cross-referencing between different panels. And again, so say for the IO 360, it's compatible with these plus products that I mentioned. These are the added in the additional 30 that you might want to make up from a panel that's already off the shelf. Okay. I think that pretty much, this is just another graphic just to say the same thing. That you get this code set, and then you can either customize everything you want from scratch. You don't have to do 800 genes at once. Maybe you're only interested in 200 genes, and that's what you want to develop your very own custom code set. Or you can kind of do a combination where you start off with 770 and can add in the 30 that might not be on your panel. And that obviously would include messenger RNA or protein. Okay, so the custom code set design, I just want to let you guys know that you would register at NanoString. You would connect with them, and if you want to talk to me further about this afterwards, or contact me, I have my card if you're interested. connect you with the field science, the field application scientist here for our area, and then just let them know what you're interested in, and they'd be able to say, okay, well, they might be able to tell you something that you don't know, which is, I've found that they've been really helpful because there's a lot I don't know, so they've been able to help. But in general, you would email them these gene regions and so forth, and then their bioinformatics team would initiate this design upon the receipt of the gene list and the sales quote. So in essence, what they would be doing is making you a code set, and hopefully if you're going to be paying for it, you'd make the code set that would last you, you know, a long time, because that code set, again, is going to have a unique barcode for each of the mRNAs that you're interested in this example. So basically what they'll return to you after that is the design report, and then the IDT order form. For people that order primers, they're familiar with the IDT company, where you just, the form would already be there and done, and you would just fill it out, and then you pay, the investigator would pay to get those oligos made, which would then be used to link to the code sets. So we're coming into the home stretch here, but basically I just thought, I went a lot into the mechanistics, but I thought maybe just to give you an overall pipeline. So basically you start off with a hypothesis. You know, you've got a question, you've got your study and your experimental design, you've got the sample quantity, you know, quality control, and basically you've decided on whatever they call them encounter or NanoString assay that you're interested in. We would process your samples here, and there are a lot of steps that I haven't gone into, a lot of minutia once you get these files, and maybe I should stop and swing over here to the left side, that you first start off with cartridge definition files. So if people deliver samples to us, we find out about their project, what you're interested in. Naming a sample is obviously really important, because that sample name is then going to be forever associated with the code set data, and you want to make it something that's unique, but not too short and not too long, sort of the Goldilocks of cartridge definition files. And then you have your reporter library file. So any code set that's ordered from NanoString, that basically is the code set keys. Like, so it includes the keys for all those unique combinations of colors for the barcode, as well as all the positive and negative controls that are included with each code set. And then at the end, after the experiment is run and you've run that cartridge, you get an RCC file. And basically that's just all the counts for all the, and this, let's just say you did a messenger RNA experiment of 800 RNAs, it would be all the counts for each and every one of those 800 genes. And so then you'd go over here, we get that file, we'd send it to you. Before we send it to you, we actually QA/QC it, so we run it on the Encounter software. And so we look at things like the imaging quality, the binding density. We look at how the positive and negative controls that were included panned out. One thing I did want to mention, like anything, the devil's in the details. One of the most, that I think for us is going to be the most challenging as we learn more about this is, yes, it's true that NanoString works very well with small amounts of nucleic acid and fragmented nucleic acid. But you've got to be able to, the investigator has to be able to QA/QC that before they give it to us, you know, so that they say, oh, we have this many nanograms per microliter of RNA. And the reason is if we have highly fragmented nucleic acid, like with FFPE, that's why we have to load 300 nanograms total onto the platform, because we want to get the right binding density of nucleic acids that we know that are going to bind to our reporter probes. So you can overload the sample or you can underload it. So for us, at least we have to have an idea of how much is going into the hybridization reaction. But once we're able to achieve that, that range, and it's a pretty forgiving range, then we're okay. What we've found with a couple of the experiments is that the RNA was of such poor quality, the folks didn't think that that was the case, and so we loaded basically what they told us they had, and what what it was. It still worked because, like I said, it can work on over 6 logs, but the binding density was a lot lower. So, so just imagine that this was, half of these strings weren't here. So it can still do counts, but what you risk here, and this is where bias for NanoString can come into play, what you risk is if your binding density is too low, if you have naturally fewer counts of a particular gene in your sample, you're not going to see that as much. So you'll have a bias towards the genes that have many, many copies. So also, again, there are a number of, when we QA, QC it, we go through the software and find out is because with all the positive and negative controls and all the housekeeping genes that come with each set, we can find out if it was an error on our part or an error just in terms of variability of the samples that were given to us. So as I mentioned, these are some of the assays that we run or that are in progress. We've, in the process of doing a microRNA panel from exosomes, as I mentioned with Megan in the back. We've gotten really nice data from a mouse myeloid innate immunity panel. Again, this was for 800 genes. And we're about to run another mouse pan-cancer pathways panel for a breast cancer investigator. And we have a couple of other projects on the horizon. If I want to now entertain questions as you have them, but also to let you know that to feel free to contact me or especially the lab, and we can find out about what you're interested in and how we can help. And thanks so much for your attention. And if you have any questions, I'd be happy to try and address them. Yes?
Thank you, wonderful talk. Just a question, what is exactly the cost?
Yeah, okay. I somehow knew that was going to be the question. So the answer is it depends, right? So like
right.
the Bible's free, but the cover's $11.95, right? For the Bible salesman, yeah. So it depends on the number of samples that you have and the codes that you use and everything, but just as a rule of thumb, so it really does depend. But as a rule of thumb, it can cost between, if you ran many samples, like over 100 samples, you might be getting into the cost of 50 cents per gene per sample up to $2 or $3 per gene per sample. So, and again, that's just like for the code sets. So we don't quite know yet because the projects that we're doing now was based on a pilot program that I negotiated with NanoString.
Okay.
So we had people apply, we had fantastic applications, but we were just doing the ones that we felt like we could get good, all of the applications are fantastic, but we could only fund 3. I think it's probably going to be in the neighborhood of about $35 per sample is what I'm guessing. So that's one answer to your question. The other answer, and you haven't asked this, but I'll answer it anyway, it's about the protein piece, because for people that are interested in proteins, if you're working with proteins that they've already developed, and they have, if you went, excuse me, I have a cold, and I'm just like, oh my God. If you are interested in one of those proteins, it's no problem. Those can be included with whatever panel or code set, especially if it's off the shelf or if you want to slightly customize it. But like I said, as far as I know, I think NanoString has somewhere between 50, might be up to 80 now because things happen fast. Proteins that they've already QA, QC'd and are ready to go. But there are proteins that I personally am interested in, and even isoforms of proteins. And what I've been told is that, okay, as long as you have an antibody that you know that works, either worked in your hands or published in the literature, then you work with us, we purchase that, and then we make you a code set for that protein. I've been told it would right now the cost is really high because it's sort of like, I guess, when we were kids, the big calculators that were like this that cost $200, and then you could get them for $10 at CVS 10 years later. Right now, to do one of those proteins is about $5,000. However, once you did it, you would have that, and that what I've been told is that the code set for that protein, it would last for 10 years or forever, you know, for a long time. And certainly by the end of that time, you know, I'm sure there'd be either new technology or it would be a lot cheaper. But does that give you kind of a ballpark idea?
Yes, so
Yeah.
just to have an idea, because I'm interested in RNA profiling, particularly microRNA or mRNA profiling.
Right.
I already used the NanoString
Right.
at the Ohio State University.
Uh-huh.
And I would like to know also if you need, because you said that you need like about 100 nanograms of RNA, more or less, right?
Or for the exosomes, it would be
For
less.
the exosome can be less. But
yeah, actually, it is a lot less, maybe 20 nanometers.
Yeah, right. But what about the quality? Like, do you measure the quality with a qubit, with another specific, you know, because the quality is, because sometimes when you have to extract from cell lines or tissue, it's fine, because the quality is always good.
Right.
But when you have to extract from exosomes or from some other samples, like fixed samples, or, you know, it's gonna be, the quality of the RNA is gonna be.
Right. So this is why at the very end, when I mentioned the binding density and that the devil's in the details, like for me, this is trying to get a really good idea of the quality before it comes to us. We're in the lab going to be getting in the next, well, definitely within the next 2 months, a qubit. We're going to be getting a Maxwell, and it's going to have a qubit with it. And so I'm really excited about that just for this reason. It's just for that reason, because sometimes you don't, I mean, it's like the only way to find out is to do the experiment.
Right.
And for a number of the applications, I would prefer to use, and this might not be, it's not going to even compute as much for, well, certainly for exosomes that Megan is working with, but for microRNAs, that's kind of its own different beast, and you probably already know about some of the caveats with that. But if we could, and I know that there's a Agilent tape station here on campus, just QA/QC to at least get an idea of the molecular weight of the total RNA going in, you know, so that then, let's just say you're just dealing with messenger RNA. So like one of these examples that they just had nanodrop data, but we could tell from their 260, 230 ratios that they had a lot of salt contamination.
Yeah, that's the problem, the chemicals.
Exactly, yeah. So yeah, all of these things are things that we can work on with you, and it's something that I'm talking to NanoString about in terms of, you know, trying to negotiate, because it's expensive. So if you're running 12 samples, I mean, typically the code set and as you know, the code sets and the universal reagents, and I'm just talking off the shelf 800 genes, right? It would be less if it were custom and you were using 100 or 200 genes that you're looking. Just to say, an off the shelf, it's about, it's close to $4,000 for 12 samples, maybe $3,600, because you buy the universal reagents, which you would have for any reaction, no matter what, and then you'd have, you know, you're buying the particular custom code set.
Okay.
But yeah, that's something that I really want to work with NanoString on, because once you have, like going into here, sorry to make stick here, once you get to the, you know, once you get to this stage here, you know, once you're committed, right? You've done the hybridization, and once you load everything onto the prep station, that's the $3,600. You know, that it's
exactly,
going.
it's gone.
Right. I mean, the good thing is if everything goes well here, and that lasts about 5 hours, you get everything on that cartridge that I sent around. You can then analyze it, and if something happens, I mean, we have a backup power supply, local, that, you know, is a really good backup, but even if this were to fail here, you could always reread it. And in fact, in theory, it's supposed to be good. We keep them cold and just save them, that you could actually re-image it, you know, a month later, 2 months later, 3 months later. But this is the key. And so, like, working with NanoString, like, you know, all of us work in different biological systems for those difficult samples. How do we figure out what data, you know, how do we figure out how much we need to load? Because we don't want to underload, but nor do we want to overload. Because if we overload, and this picture here is chock full of colored strings, then you're not going to get accurate data. It is, however, that binding density is very forgiving, which is really nice. You know, it goes from, you want it above 2 and say less than 200. So it's actually a pretty wide forgiving range of binding density.
Thank you.
And so again, I have cards here if you want them. No pressure, but I'm happy to talk to you about it, and also happy to tell you there's a lot I don't know and I'm learning too, but I can hopefully get you the answers to your questions if I don't have them. Thank you.
Thank you.
Thank you.